Good morning, all. Let's get started. Are there any questions before I start the lecture? Okay, so a couple of reminders. Quiz 8 is open and it's due on Friday, the day after tomorrow. Quiz 9 opens, I didn't correct the typo. Quiz 9 opens on March 20th and it's due on Wednesday, March 24th. So March 24th is not a Friday, March 24th is Wednesday. Just mentally cross this out. And there is a participation assignment that is due today. It's not open yet. It's going to open right after the lecture. So don't forget to take the participation assignment that is due before midnight tonight. So today I'm going to continue with the forces of evolutionary change, and we are going to wrap up the topic of forces of evolutionary change today and move on to the next one, speciation and extinction, on Friday. All right, so last time I left off on Hardy-Weinberg equilibrium, which is again at some hypothetical state of the population over time that remains constant. So Hardy-Weinberg equilibrium is theoretical state. And it's calculated as, last time I talked about population of ferrets and I focused on this example here on the population, um, we were looking at the alleles that encode fur color and the population of ferrets. So there are two fur colors, the dark one and the tan one. And the two fur colors are encoded by two alleles of the same gene. So capital D is dominant and it stands for dark fur, and lowercase d is recessive allele and it encodes the tan population if an individual is homozygous recessive. Um, let me ask you again what I asked you last time. Um, like, I would like to calculate allele frequencies for some subpopulation on this picture. Let's take the three ferrets over here. This kind of question is going to come up on the exam, so I want to make sure that you guys who attend, at least, that you got it down. So three ferrets over here. What is the frequency of the recessive allele, the allele that encodes for lighter tan fur color?
Is it 50%?
Oh, hold on. I have my volume too low. Say again?
50%.
Yeah, that's exactly right. Would you mind walking me over how you calculate it?
Yeah, so there's 3 recessive alleles out of 6 total alleles. So you
Mm-hmm.
just do 3 divided by 6.
That's exactly right. Nice job, Christine. So, as Christine said, first, we calculate the frequency of the recessive allele in a population. And let's imagine we're looking at the population of 3 ferrets here. So 1, 2, 3. And to calculate the frequency, you divide the number of alleles of interest um, over the total number of alleles. So total number of alleles is 1, 2, 3, 4, 5, 6. It's easy to sometimes people confuse like the number of alleles with the number of genes. Okay, so like it's kind of tempting to count this as one, this as two. It's tempting to count individuals, but it's important to remember that we're counting the alleles, and each individual ferret will have two alleles, one inherited from male parent and the second one inherited from the mother. I'm sorry, yeah, father ferret, one inherited from the father ferret, and one inherited from the mother ferret. Okay. Okay, let me check the chat here. And everybody is correct. Nice, very nice. I'm glad you guys are getting it. All right. So, and just a reminder, we can in order to calculate the theoretical state of Hardy-Weinberg equilibrium, theoretical frequencies of genotypes in a population, we first have to calculate the frequency of alleles. Let's say a gene has two alleles. I'm not going to make it complicated and give you like six alleles for the gene or anything. So we're going to be working with just the two alleles for the same gene. And it's kind of nice because the frequencies of the allele that you did not figure out computationally can be calculated as 1 minus whatever, or 100 minus whatever, if you're working with percentages instead of fractions. So in the previous example, the frequency of allele for the whole picture, if we take the whole population here, all of the ferrets instead of the just subpopulation of 3, if we take all of the population and calculate the frequency of the capital D allele encoding for the dark fur, we would have zero point 0.6 or 60%. So 60% of all alleles are going to encode for dark fur. And the frequency of the alleles for tan fur correspondingly is 1 minus 0. 0.6, or 100% minus 60% is going to be 40%, or 0.4. Now, we also went over how to calculate genotypes, and to calculate genotypes, you can do either, there are two methods, either through Punnett square or through the equation here, which is basically the same thing. Just one is more visual and another one is more computationally oriented. So in order to, if you choose to calculate the frequency of the genotypes using the frequency of alleles, uh, from using the Punnett square, it's important to remember to mate two heterozygous individuals. Okay, so your female is going to be heterozygous and your male is going to be heterozygous. And you just multiply these gametes by each other and you get various all possible genotypes for the offspring. So homozygous dominant, homozygous recessive, and heterozygous, meaning like occurring twice because we have two heterozygous parents. Uh, okay, and to calculate the genotype frequency, you basically multiply the frequencies of individual alleles. Okay. Any questions about that? Is that all clear? I'm spending a lot of time on this, but that's because it was, this kind of concept was confusing in the past.
I have a quick question.
Yeah, go ahead.
Is the genotype frequency only between like two gametes, or like how would we do it for the example that we just did for the allele frequency?
Right. So let's say the example, you mean with three individuals in a population?
Yeah.
Okay, so we had 50-50 frequencies for dominant and recessive alleles. So double D would be like the genotype, the homozygous dominant individual would have 0.5 times 0.5, so 25% would be the genotype frequency for the homozygous uh, dominant individual. For the homozygous recessive individual, it would be also zero point 0.5 times 0.5 because the frequencies of dominant and recessive allele are exactly the same. So you would work with whatever frequency you're calculated. So it would be again 0.5 times 0.5, 0.25, or 25% of individuals would be, is the accepted frequency or expected occurrence of the heterozygous, well, 0.25 times 2. Uh, so 50% would be the expected occurrence of the heterozygous genotype, heterozygous
So you can
individuals.
only do it between two individuals.
Uh, it's it's not necessarily. We're not working with individuals. We're calculating these occurrences for the populations. We're just like calculating the theoretical occurrences in the populations under Hardy-Weinberg equilibrium assumption. So in order to do that with a Punnett square, we're just crossing the heterozygous theoretical individuals in order to calculate the actual percentages
Okay.
occurrences of the genotype. So where these individuals are theoretical, what's important here is the actual occurrences of the alleles. And the what we get as a result is the occurrence of the genotype. So 36%, in this case, 36% of all individuals in, oops, in this picture here would have the genotype uh, of double Ds, or would be homozygous dominant. 24 times 24.24 times 2, it would be 48. So 48% of individuals in this picture would have the uh, would be heterozygous for the fur color. Okay, so if you're calculating the frequencies for the genotypes, it will be genotype frequency. If you're calculating the frequency for individual alleles, it would be allele frequency. I gotta threw a lot in there, but was any of that useful, Christine? Okay, I see
Yeah,
your
I kind of
message.
just had a brain fart, so
That's
I got
okay.
it now.
Anyone else?
In your yeah, in your like final answer, would you say the probability for capital D, lowercase d, you would add those two percentages together, 24 plus 24 equals 48? I
Yeah,
think you said that. Okay, I just wanted to make sure.
yeah, that's
Thank you.
exactly right. Occurrence of heterozygous individual is counted twice in the Punnett square, or should be counted twice.
Okay.
And this addition here is represented by this term in the equation for genotype frequencies, 2 times PQ. And this is basically to account for uh, heterozygous individuals occurring twice in the Punnett square. All right. So in real populations, the assumption of Hardy-Weinberg equilibrium are always violated, meaning evolution is taking place. So what are the assumptions of Hardy-Weinberg equilibrium? Again, is that there is no natural selection occurring. Uh, Individuals do not emigrate or immigrate from in the population. There is no sexual selection going on. There is no what else? Yeah, individuals do not mate at random, meaning there's no sexual selection going on. And another big assumption is that the alleles do not mutate. The genes do not mutate, which they do. They can mutate randomly. They may not be selected for even, but as I shown and as I showed in a couple of lectures ago, like once in 25 millions of years, maybe the gene may mutate, in which would lead to new phenotype. Um, so natural selection and mutations do occur, and that's why real world populations do not remain in the Hardy-Weinberg equilibrium. Random changes in allele frequencies are common. Individuals rarely mate at random. It's true for us, it's true for ferrets, it's true for anything basically that is not plant or a sponge, living in the sea, or those kind of organisms. There's always some level of a migration between the populations. So in the next couple of slides, I'm going to go deeper into each of these reasons why Hardy-Weinberg equilibrium is not maintained in the real world population. So the first reason is the natural selection, and it can shape the populations. There are three types of natural selection uh, that have distinct effects on the phenotypes in a population. The first type of natural selection is directional. Second type is disruptive, and the third type is stabilizing selection. So within a directional selection, the phenotype moves, well, literally in some sort of direction. So the example here deals with the changes of color of moths. So let's say in some forest where the trees have lighter colored bark, we will see a certain distribution in the colors of the moths. So the x-axis here would be the increasing pigmentation. Like imagine some sort of metric for how dark the moths are. And on y-axis is number of individuals. So the bell curve here represents the frequencies of the color of the color of moths within the population. So we have lower number of individuals that are light, lower number of individuals that are dark, and the highest number of individuals have the medium color that matches closely to the color of the bark. Now, let's say the forest underwent succession, and one species of trees were kind of pushed out by another species of tree. So, like the another species of trees was more competitive compared to the original one. So they gradually overtook the plot of land. And what's going to happen with the moths? So moths are going to gradually change their color to the new color of the bark. Like over generations, the color of the offspring will gradually change, becoming darker to that would match the new color of the tree bark. Can anyone hypothesize, or can anyone guess why this would happen?
It's because of the predators, isn't it? They would hunt on the lighter colors, meaning the darker ones would survive and pass those genes on.
That's exactly right, Madeline. Very good. So the moths that stand out, that are more visible, like on the contrast, would be eaten because the predator can see them. But the moths that can blend in well with their surrounding environment, they have lower chance of being eaten and the higher chance of reproducing. And remember, what is considered evolutionary fit, like fitness in terms of evolution, is basically the ability of an individual to survive and survive long enough to reproduce and be successful in reproducing. Any questions about this example? And this kind of change in phenotype is called the directional natural selection. Directional selection. It seems common sense, right? Well, I would hope so, at least. Now, disruptive selection is interesting, and it occurs in many aquatic habitats. And how it works, I'm going to explain how it works on the example pictured here. So imagine you have a habitat of pebbles near some stream shore, and the pebbles, let's say, are uniform in color, and on the pebbles, pebbles are the habitats of for snails. And we have the graph here shows the frequency or occurrence of the color of a snail's shell. And on x-axis here is again some metric of color, which depicts basically from lighter color to the dark color as the axis moves to the right and the right. And the y-axis is the number of individuals. So some lower number of individuals will have light color, some lower number of individuals have dark color, and the highest number of individuals will have the medium color that would match the pebbles. Now imagine there is some sort of disturbance going on, like there was a flood, and a whole bunch of pebbles from upstream were imported to the habitat of these snails. And these pebbles from upstream are going to be much lighter. So now in the habitats there is a mixed color, mixed pebbles. Some are dark brown, and others are light tan color or almost white. So what we will have over many generations, we will see disruptive selection. We will see that more higher number of individuals are going to be lighter colored, and higher number of individuals are going to be darker colored, while number of the medium colored individuals that used to match the old pebble color is going to plummet. So this sort of movement of the phenotypes of individuals within a population to the extremes with respect to some metric like color of the shell is called the disruptive selection. So disruptive selection is when your phenotype moves to the extremes of the spectrum. Does that make sense? I see Dylan nodding. Anyone else? Dylan, thank you for turning your
Yeah,
camera on.
it's pretty easy to respond when you can just shake your head.
All right. Uh, Okay, thank you, Alexander. Thank you, Farouk. Yay. So the third type of selection is the so-called stabilized selection, and it favors intermediate phenotypes. So it favors the highest occurrence within a population, whatever metric occurred the most frequently within a population. So the example here deals with the birth weight of children. So let's say many years ago, children that were born had a bell-shaped distribution. So x-axis in this case is the birth weight, y-axis is the number of individuals. So lower number of individuals are born with low weight, low number of individuals are born with large weight, with heavy weight, and most of the babies that are born have some average weight. Okay. Now, over time, the stabilizing selection would narrow the width of this bell shaped curve. And why would that occur is because babies with that are born with low birth weight as well as very unusually high birth weight, they have higher probability of developing diseases and not surviving to the reproductive stage and not reproducing and passing on their genes. Okay, so that is why, and that is the underlying mechanism of the stabilizing selection. It's basically narrowing the in the stabilizing selection, the bell-shaped curve is becoming more narrow due to lower survival rates of the individuals uh, on the extreme ends of the spectrum, on the extreme ends of the bell-shaped curve. Does that make sense? All right, Christine, good. Awesome. Now, this is like common sense and all, and we understand that if the allele maybe is causing some disease, it may be gradually eliminated because the person will die from the disease and would not reproduce. Well, not necessarily person, but let's say individual within a population. So if natural selection is so awesome and kind of weeds out the unfit individuals, why would we have alleles with harmful, which cause the diseases with some non-beneficial effect on the population? And usually, well, interestingly, it's because some of the diseases are actually beneficial for survival of the population. Let's go back to the example of the sickle cell disease, which is caused by mutation within a gene that encodes the protein called hemoglobin. So again, hemoglobin is a protein in the red blood cell that carries the oxygen. And the misshapen hemoglobin molecules, they stick together, and the stickiness, it causes the abnormal shape of the red blood cell. So as a result, the red blood cell does not carry as much oxygen. So the people who are homozygous recessive, only those people will have the sickle cell disease. People who are heterozygous, they're not going to experience the symptoms of the disease. Okay, they will have normal functions. But, well, not but, and interestingly, people who are heterozygous for the sickle cell disease, and who populations that are most frequently are sick with sickle cell disease, are located in the same regions where malaria is spread. So malaria is caused by a parasite. A parasite, it's a kind of organism that is called a protist, and the name of the protist is called Plasmodium. So Plasmodium lives within it's carried around by mosquitoes. So Plasmodium lives in blood. So if a mosquito lands on an infected person and drinks some blood, then and it carries this blood to another person, basically feeds on another person, it transfers the plasmodium to the uninfected person, and this person can get sick. So the malaria is uh, is caused by this plasmodium, and it's unpleasant disease. It's hard to get rid of it, and it causes like very unpleasant symptoms. Like you get fevers, you're sick, and when you think you're cured, you get sick again, and it's just a mess. Used to be treated with quinine. Now it's well, still in some countries it's treated with quinine, but there are more effective treatments now. Anyway, I keep diverging from the subject. So, in parts of the world where catching malaria is common, basically sickle cell alleles are more frequent in the population than malaria-free parts of the world. Uh, I'm looking at the chat here. There's a question: does this relate to anything of heterozygotes being more genetically diverse, which increase survival of species organisms? Not quite. So in the next slide, I'm going to go over it. So sickle cell allele confers heterozygote advantage when malaria is a threat. So heterozygotes for the sickle cell allele do not have the sickle cell disease and are protected against malaria. In two heterozygotes, if two heterozygotes mate, their child might have sickle cell disease. So basically, a lot of people who live in the areas where malaria is very widespread, they a lot of people are heterozygous for the disease, for sickle cell disease. And basically, that has to do with Plasmodium not liking some of the cells that have misshapen erythrocytes. Plasmodium feeds on blood, okay? Plasmodium loves blood. And if some red blood cells are poor in oxygen, Plasmodium will not survive. In heterozygous individuals, like they have very like some proportion of the red blood cells will be misshapen due to this recessive allele for a sickle cell disease. So because of that, these people who are heterozygous, they are not going to kind of transmit the malaria further because Plasmodium will not survive in their partially oxygen poor blood. So a heterozygous individual does not feel the effect of sickle cell disease. A small portion of the red blood cells are going to be misshapen, but the small portion is still enough to prevent Plasmodium from survival and from being carried forward by mosquitoes to the healthy individuals. That was kind of long-winded explanation, but did that make sense to people? Not as active. What kind of questions do you guys have about it?
Are the people with the double recessive sickle cell disease, are they even more resistant to malaria? And also, like, is it, I don't know how severe sickle cell disease is. Like, is it like if you were double recessive, like would it cause you issues? Could you not, do what you wanted? Okay.
Yeah, like I would bet people with sickle cell disease would not care about malaria because they would have bigger issues to
Yeah, okay,
to worry
okay,
about.
okay.
Yeah, so
I thought that.
it increases the chance of strokes, and because we need oxygen everywhere in our body, if the erythrocytes, the red blood cells, do not carry as much oxygen, there are a whole slew of issues that are stemming from that, like and weakness and fatigue is only very minor of them,
Yeah,
all of our
I got a small taste of low iron in my blood after I donated. So now that I'm putting two and two together, although they're different, yeah, no, having low oxygen level, low iron level in my blood was not a nice experience.
right. Okay, any questions about the heterozygous advantage? How come homozygous dominant have a high chance while recessive means you have it? Lauren, can you unmute yourself and rephrase your question? I'm not sure I understand it as is written.
Yes, so I'm looking at the a zygote dominant where it's saying the high susceptibility to malaria. And then I'm looking at the recessive, which says you have the sickle cell disease. So if they're both homozygous, why is one more susceptible to getting it and one means like that you have it if you have like the recessive alleles?
So this one, are you asking why is this highly susceptible, but this one is not?
Yes.
Okay. So we're working with two things here: malaria and sickle cell disease. Okay, so malaria is caused by a parasite. Sickle cell disease is caused by genetic mutation that causes misshapen red blood cells. So if you have healthy red blood cells, healthy and plump red blood cells, Plasmodium, which is the cause of parasite that causes malaria, it loves the red blood cells. Okay, it loves these plump, healthy red blood cells that homozygous dominant individuals have. Now, sickle cell disease is caused by a recessive allele, and the homozygous recessive individuals for the sickle cell disease, they develop sickle cell disease, and their blood cells are not as plump and full of oxygen that Plasmodium prefers. So it's just Plasmodium does not survive in these type of blood cells, in these sickle cells. Does that make sense, Lauren?
Yes, thank you.
All right. Anyone else before I move on?
So it's beneficial for malaria to be recessive, but it's better overall to be heterozygous.
Yeah, in the locations where malaria is prevalent. So it's an adaptation on a population level. It's an adaptation to the environmental conditions. So to prevent malaria, not to prevent, but because malaria killed off a lot of individuals who are homozygous dominant, heterozygous had the greater chance of survival. So there's a lot of high prevalence of heterozygous genotype within the areas with high occurrence of malaria.
Okay, makes sense. Thank you very much.
And the sickle cell disease is just a kind of a byproduct of that, the disadvantage of having lots of heterozygous individuals within a population. Okay, moving on. Now, sexual selection. Sexual selection directly influences reproductive success, and it's a type of natural selection. It results from variation in the ability to obtain mates. Okay, this can include like competition of individuals for access to a mate, like rams and the beetles, or it can sexual selection can also occur when a female chooses a male, like this weaver bird. This is a male weaver bird occurring in various places across African continent, mostly below Sahara because it's a desert. Very few things occur there. So this male weaver bird, it's as a part of attraction of a female, it builds a nest, and females fly around, look at nests, and choose whatever nest looks best. And this is how females choose male weaver birds. Okay. What else? Well, that's basically it. So there's various types of kind of sexual selection strategies. Sometimes a male will, like in peacocks, male will have this exquisite tail that attracts females, and this is another sexual selection strategy. Okay, so moving on to another reason why Hardy-Weinberg equilibrium does not occur in the real populations is mutations. So mutations cause evolution to occur and Hardy-Weinberg equilibrium to not be maintained. Mutations create genetic diversity. There are beneficial mutations that if the mutation is beneficial, then it's passed on to the next generation. So the frequency of the beneficial mutation is increasing over time, over generations. So a straightforward example for that is basically the development of the antibiotic resistance bacteria. Let's say we have a population of bacteria that over time generated some random mutation. that is responsible for antibiotic resistance. If we introduce antibiotic to a bacterial population, the bacterial population over generations in the presence of the antibiotic is going to become antibiotic dominated by the antibiotic resistant individuals. However, if we leave the bacterial population alone and do not introduce antibiotic, the population will still increase, but the number of the antibiotic resistant individuals are going to remain the same. Maybe, if it's been well, yeah, there's going to be some small number of individuals there because bacteria do not have sexual selection. It's kind of they have asexual reproduction. Another reason why the Hardy-Weinberg equilibrium does not occur in real life is genetic drift. And genetic drift is basically the sampling error within the population. It can be interpreted as sampling error within the population. So sometimes by random, only some individuals in a population reproduce, causing a change in allele frequency to occur purely by chance. And in this example here, we don't work with the live things. Like the sampling error is visualized by sampling from a jar with two colored balls. Okay, so we can start with, and the balls represent the allele frequencies. So let's imagine that we have a population with allele frequencies 50-50 for a particular gene, for allele frequencies for a particular gene 50-50. But let's say only some individuals, only a portion of these individuals reproduced and another portion did not reproduce. And then the individuals that reproduced, basically their allele frequencies were set 70 to 30%. Now, if this new allele frequency is going to be our like long-term frequency just due to some sampling error from the original population, we can have another sampling error, some sampling error, basically another random sample of individuals who will reproduce and pass on their genes. And these individuals who reproduced will produce like a generation or a population with 90 to 10% ratio of the alleles for some gene. And some further sampling errors can rarely result in like 100 to 0 ratio of allele frequencies. So basically, there may not be any sexual selection going on, but just from the small sample of the individuals that reproduced, we can have drastic changes of the ratios of allele frequencies in the following generations. It might be it might become more clear when I give this example here. So there's something called a founder effect that causes genetic drift. Let's imagine we have like some genetic diversity in the original population. Let's say a portion of population has left the old population and emigrated into a new habitat. And because these few individuals will not represent the whole genetic diversity within the population, the new population will be much less genetically diverse. The new population will have no access to the green allele here or no access to the purple allele here. So the new population will have only blue alleles, orange alleles, and red alleles. Okay, so the picture here shows basically illustrates the Ellis von Creveld syndrome, which is very rare. Ellis von Creveld syndrome was originally described in Swiss populations. It's populations like in Swiss Alps, I think. And it appeared in the United States after 200 followers of Amish denominations, they immigrated into the States. So within Amish culture, it's very common to not seek mates within their own population. And because there were no external like alleles that were introduced for the same genes, because Amish people like looked for spouses within their own population. This disease, Ellis von Creveld syndrome, it became more prevalent within the Amish population. So the Ellis von Creveld syndrome causes dwarfism, extra fingers, and some other symptoms, and its prevalence among the old Amish population in Pennsylvania is 7%. And this is crazy high for compared to population outside of that region. Okay, and it's basically a result of intermarriage and marrying people from their own populations. So this is the founder effect. The founder effect is basically uh, is found when the original genetic diversity is reduced by a portion of population like immigrating, and this portion of population having much lower genetic diversity compared to background population. Was that was that clear? Oh, Christine, I see your question from before. Is being anemic genetic? It depends on the cause of the anemia. Like there's no one answer for that. Like anemia can be caused by various things. So sometimes it's nutritional deficiencies, and sometimes it can be genetic. Yeah. So moving on. Population bottlenecks basically cause genetic drift. So remember that one of the causes why Hardy-Weinberg equilibrium is not true is through genetic drift, and genetic drift is often characterized by the bottleneck effect. Population bottleneck occurs if the size of a population is greatly reduced and genetic diversity is lost. So in the picture here, let's say these colors here represent the genetic diversity of the population of, oh, I'm blanking what that is called in English. Anybody? What's the name of this cat here? Leopard?
Cheetah.
Cheetah.
What?
Cheetah.
Cheetah.
Yeah.
Thank you. Yeah, cheetah. Thank you. So let's say this picture here represents the genetic diversity of for some gene in cheetahs. But these cheetahs, let's say it was hunted for the fur and for sport, and it was hunted down to very like few individuals, but then conservationists intervened, the government intervened and said, hey, we want our kids to know what cheetah is and to be able to see cheetahs 20 and 30 and 50 years from now. So let's start conserving these individuals, and the government may introduce legislation banning the hunting of cheetahs. They may introduce like legislation to maintain the populations which cheetahs prey on, so the cheetahs are happy and reproduce and feeling good. So they kind of, let's say they succeed, but because the population of cheetahs was reduced in the first place, the genetic diversity of cheetahs was reduced too, because the individuals that remained may had like alleles that are represented by purple, blue, and red. So the new population of cheetahs will have much less possible alleles for a particular genes compared to the original one. And because of that, because of the reduction of the genetic diversity, there's higher risk that some extreme event or some disease will wipe out all of the population of the cheetahs. Now I have a question for you. Can population of cheetahs ever recover genetic diversity? Are there any mechanisms that you've learned about so far that would allow recovering the genetic diversity of cheetahs, theoretically?
Dramatically. I mean,
Theoretically, that was not dramatically. Yeah.
okay, I
Theoretically.
was gonna say, once again, the mutation over thousands of years, collections of other random events that eliminate cheetahs, but that would even reduce genetic diversity more. That's you would almost have to rely entirely on mutations, or is there another mechanism that I can't recall?
based on what we've learned so far, mutations it is. And I see Nicholas posted his
Yeah,
response in chat. So mutations is correct, but it's going to take much longer time, right? Mutations occur. It's mutations that stay in the population. They occur not frequently at all. So the example I gave before gave a number of 25 millions of years. So it's much more probable that some extreme event will further reduce the genetic diversity of the population that was endangered before, or rather than unless we're really good, really good at management of populations that are endangered. So this is kind of an illustration of why conservation is important and why like early conservation, while the population size is still big, is important, you know, because once the population size is below the subcritical threshold, then there's going to be a high probability that genetic diversity cannot be recovered. Like that genetic diversity will be greatly reduced, and this will increase the susceptibility of populations to some extreme events. Now, non-random mating causes evolution to occur. So this is another reason. Non-random mating is the reason why Hardy-Weinberg equilibrium does not hold in the real population. So mating is rarely random. We have sexual selection, and I already touched on that plenty in the previous lectures. Like we, as people, choose individuals who with symmetrical facial features and symmetrical body features in general because it's associated with better health. And there's like some other individuals within other species, they have various strategies like this. What is it? It's not a frog, it's a toad. It's a toad. So the toads sing, the male toads sing to attract the willing female. What else? The example I gave a couple of slides ago was with weaver bird. The male weaver bird builds nests in order to attract female. So so mating is not random. That's the bottom line. Now, can you think of any organism that has random mating? Do you think there's random mating in nature?
Press the wrong button. Could you say I can't. I guess I'm gonna use salmon as the example because don't they just kind of like lay their eggs and then they're kind of just they inseminate the water and then just kind of randomly you get a sperm, you get an egg, boom, there's a egg baby.
That's a great example. I see Lily gave an example of plants, and that's a very good example. Like plants just grow there, and if the plants are pollinated by wind, which a lot of them are, then wind does not know how to select a plant, right? Wind just blows, and whatever pollen got picked up into or whatever plant it's landed on is going to be random. So plants that are pollinated by wind, they have random mating. Bees, I'm not sure if bees are bee reproduction. Inacy? So can you unmute yourself and expand on that?
Sure. So to my understanding, there's like a queen bee, and then all the male worker bees just like fertilize the eggs or whatever. So like she doesn't really get to choose her mate.
Okay, that might be the example. I don't know much about reproduction of bees, but from what you described, that would be accurate. That would fall under the non-random mating strategy. Okay, so the last slide, we have 1 minute. And gene flow causes evolution to occur. So lateral gene flow is basically caused by individuals within a population moving, like emigrating or immigrating to a population. So gene flow moves alleles between population and increases genetic diversity. And this can change the allele frequency in both population. And the picture here shows two populations of rabbits. one population having prevalence of lighter fur and another having darker fur, and these rabbits may not like be separate from, if populations may not be separated from each other for long. Let's say they used to be separated by I don't know, a river, but then the river dried out and the rabbits started mixing with each other, and the genetic diversity would increase and gene flow would because of the gene flow, lateral gene flow. All right, so that's it. And are there any last minute questions?
I just have a quick question about the participation assignment today.
Yeah.
I realized that it hasn't come up on web courses yet, so I was wondering when that'll be released.
Just in a second after I finish the lecture.
Okay, thank you.
Alexander asks, is intelligence related to genetics? That is a great question. So there are, this question, this kind of research question is answered through twin studies. Like psychologists in particular were very interested in this question, and they compared the intelligence of fraternal twins versus homozygous twins. Homozygote twins? Anyway, identical and fraternal twins. And these studies are often associated with some ethical concerns. So now they're not conducted much, but in the 50s and 40s and 60s, the studies were basically, the gist of them was in separating fraternal twins and separating identical twins at birth and placing them in different families with different backgrounds. And so they found that basically through these studies, the identical twins placed in different backgrounds, like kind of pointed at intelligence being inherited. Like no matter the environment in the where identical twins were placed in, they seem to be like at similar level of intelligence, no matter what kind of education they got. With fraternal twins, it was, I'm not going to touch fraternal twins because to answer this question, kind of identical twins are more important. But there's quite a lot of debate on that still because studies were not perfect. And there's big debate on nature versus nurture in intelligence. Like there's definitely some genetic component, but it doesn't mean if you have bad genes, you cannot be intelligent. Like I wish it was a clear cut. There was
It's
a
like
clear
you
cut
could
answer.
it's like put it this way, is like you can say I know a child's going to be a genius and they lack primary socialization, so they never grew up with a family, they grew up in a wild way, grown up by dogs, something random, that child will never be smart. It's such a complex question, and I took sociology a while ago, and I love doing learning about nature versus nurture, because like it was like, yes, nature matters because that's your attitude and a lot of that, but really if you want to learn things like physics and concepts, is you need to go to school, period. Like you won't be smart unless you go to school.
That's true. So Alexander has a follow-up is that's kind of scary, but I heard that intelligent is taken from the mother. Is that true? I think I saw like an article published not so long ago about it, but these kind of study, like I think the article that I saw was not done by biologists, it was done by psychologists, and psychological studies are riddled right now. There's a reproducibility crisis in psychology because of over reliance on statistical techniques and not having like robust genetic component to their studies. So I wouldn't put too much weight on that as of now, but I would need to actually let me check back with my partner. He's done a lot of has a psychology degree, and he knows more about it than I do, and get back to you, Alexandra. Okay. Um, Yeah, Christine says, first gen student here, so I don't get my intelligence from. It doesn't, okay, if your parents did not go to school, it doesn't mean that they're not intelligent, okay? There's lots of other reasons of not going to school, like not having money or not having parents to support you if you suddenly, drop out or not encounter a problem during school. So like, it doesn't, if a person didn't go to college, definitely does not mean that they're not intelligent. Okay. All right, guys, it's 10:24, so I'll let you go.